Hello, everyone, and welcome to our expert briefings. I'm Dr. James Beck, Chief Scientific Officer of the Parkinson's Foundation. Our expert briefing today will be centered around Parkinson's disease and how the gut is connected to the brain. While stomach or intestinal distress can lead to anxiety or depression, gut-brain connections go much further. New research strongly suggests a link between the gastrointestinal system and Parkinson's disease. Today, we will learn a lot more about the gut's potential impact on symptoms and progression. But before we begin with the formal briefing, let me just share a little bit about the Parkinson's Foundation. The Parkinson's Foundation is a nonprofit focused on bettering the lives of those living with Parkinson's through improving care and advancing research. Importantly, everything we do is done in close concert with our community to ensure that our actions are aligned with the needs and priorities of those living with Parkinson's disease. We are focused on improving care for those living with PD, advancing research towards a cure, and empowering our global community. Today's briefing is an example of just how we reach our goals. But before we do that, I want to just highlight what's going to happen next Wednesday on October 18th. We're hosting a special program designed for people living with Parkinson's who've not shared their diagnosis publicly. As we all know, this is an issue, and we'll highlight the challenges many people face when deciding how and when to share their diagnosis. You can learn more about this webinar at our website, www.parkinson.org/PD Health. So as our next step, what I would like to do is just get a chance to know who's on the call today. We've got a lot of people today, and there's a poll that just appeared. And so go ahead and select whether you're a person with Parkinson's, you're a scientist, a researcher, you're someone else who's joined, or you have a friend or family member with Parkinson's disease. This is really helpful for us to get a sense of who our audience is today, and also for our speaker. So they can have a sense on who's on the call and make adjustments to their presentation. So let's see, give a few more seconds, and then we'll see what the poll comes back as. Great. Not surprisingly, a lot of people here with Parkinson's and those who are partners of those living with PD. Fair number of healthcare professionals too, which is great, and scientists and researchers. And I think we're gonna have a great talk today that I think will cover a lot of ground and have something for everybody. So for your convenience, I just want to highlight that we are recording this episode. And so if you've missed this, or you need to step away, or you have friends and family members you think who could really benefit from watching this episode of Expert Briefings, know that it will be made available at our website. And we'll also be emailing a link of the recording and other resources related to today's topic to all those who registered for today's briefing. So now I'd like to go on and introduce our expert presenter, Dr. Carly Rush. Dr. Rush recently became a medical science liaison in medical affairs research at Abbott Nutrition, specializing in adult therapeutics. Before that transition, she was an instructor in nutritional sciences and a neurology dietitian at University of Florida Health Norman Fixell Institute for Neurologic Diseases for many years. Dr. Rush completed her master's degree in food science and human nutrition and completed her dietetic internship at University of Florida. During her training, she was involved in the research studies of a number of topics on gastrointestinal dysfunction, inflammation, pre and probiotics, and the gut microbiome. Dr. Rush earned her PhD in nutritional sciences also at University of Florida, where her research investigated the effect of the Mediterranean diet on improving constipation and intestinal inflammation in Parkinson's disease. Dr. Rush's work appears in numerous peer-reviewed journals and has been invited to present her research and nutrition-related topics at both state and national conferences. Her clinical experience spans a variety of disease states such as cancer, gastrointestinal and neurologic disorders, malnutrition, and critical care. Dr. Rush, it's a real pleasure to have you, and thank you for sharing your time and knowledge with us today.
Perfect. Thank you, Dr. Beck. I am really honored to be invited as a guest speaker for the Parkinson's Foundation for these expert briefings. I've had a long relationship now, I think, with the Parkinson's Foundation, doing some more local work and speaking. So it's, again, really honored to be invited to chat with everyone here today. And just taking a look at the number of people that we have so far, up to 850 people and growing. This is amazing. Welcome. Before beginning, I just have a couple disclosures that I do want to share. As you heard, I am now an employee within medical affairs and research at Abbott Nutrition. And while the program today is not intended for any individual medical advice or continuing education for healthcare professionals, I hope that you will find that it is an evidence-based review of the literature on this topic. Here are my objectives for today. So we're going to talk a little bit more about how that gut microbiome is altered in someone with Parkinson's disease and discuss the role of that gut-brain axis and how diet may impact Parkinson's symptoms and gut health too. And before beginning, I just want to share very briefly with you a little bit of my background in Parkinson's. So it was approximately almost 5 years ago now that I was invited to join the Norman Fixel Institute for Neurological Diseases at the University of Florida. I've done a number of work with them, partnering with their Parkinson's Foundation Symposium that was locally held in Gainesville, Florida. I saw numerous patients in our outpatient clinic that you see there with my photo in the middle. Maybe some of my former patients are and caregivers are here on the line, maybe some clinicians that I've worked with in the past too. And then of course, like I mentioned earlier, I have given multiple talks for the Parkinson's Foundation on diet and Parkinson's disease. So let's talk a little bit about the gut microbiome and the gut-brain axis. And I will preface my talk today. I know there are a lot of people with Parkinson's, care partners on the line, and we also have a really great number of healthcare professionals and researchers too. And so what I do want to say is that the content of the program today is probably going to be a little more on the technical side, but with my experience with people with Parkinson's and the care partners, you guys are all a very smart individual and group of people. So I hope my job today really takes a complex topic with a lot of nuance and simplifies it down together. All right. So now, before diving into the meat and potatoes of my talk today, I want to start off by sharing this image. So this is a cross-sectional image of the colon in a humanized mouse model. So that blue fluorescent color is a stain that shows both food and tissue. The yellow represents the mucus in the colon, and purple is going to be that bacteria. This paper was published in 2017 in the Proceedings of the National Academy of Sciences. And it was an important paper because it captured the structure and relationships between bacterial microbes and its hosts. These relationships are significant because we know our resident bacteria are more abundant than ever, even our human cells too. The genetic capability of our resident bacteria is enormous. And even though I'm showing you an image highlighting bacterial microbiota, we're beginning to learn even more about other members that reside in the microbial community, such as fungi, which we call the microbiome, and viruses even too. But what I really enjoy about this image is when you magnify it a little bit further, you can really appreciate the ability of our bacteria to break down food from the diet that I have on the photo here on the right. So what is the microbiome? I always love to start this conversation off by just giving some definitions because we use a lot of these terms interchangeably. So I think it's really important to set some groundwork too. So the microbiome, when we hear that, that's referring to the totality of microorganisms and their collective genetic material present in the intestinal tract. We know that the microbiome can really span from the mouth all the way down to the colon. The gut microbiota are microorganisms of the intestinal tract, and these could be both bacteria, viruses, fungi, and even protists too. When we think about the gut microbiome, we often think about the colon, right? And that's because we have the greatest density and diversity of bacteria in particular located in the colon. And for the purposes of today's presentation, I'm going to primarily focus on the microbiota composition and metabolism that's located within the colon. But certainly, especially within Parkinson's disease, there are some papers out there that do refer to the microbiome in the oral cavity even too. So what is a healthy microbiome? And we might want to first consider that too when we're talking about this type of topic. And it's really something that researchers have debated and really struggled to specifically define, even someone like myself. However, it's generally accepted that a healthy microbiome is a diverse one. We see lower diversity is associated with different disease populations. We can measure diversity within individuals called alpha diversity by measuring the number of species present called richness. And this figure that I have here on the left shows richness of samples from people with IBD, including ulcerative colitis and Crohn's disease, compared to healthy controls. So you can see that blue line, that as we sequence more bacteria, people without irritable bowel disease have higher number of species present than those with ulcerative colitis or Crohn's. And when we quantify this, you can see that there is a trend towards lower diversity in people with Crohn's compared to healthy controls, which I have in that little shadow box here at the top left there. We can also show diversity in a 3D space too, and compare diversity between different populations. This is known as beta diversity. Each dot that you see here on the right is an individual, and the closer those dots converge, the more similar they are to each other. So you can see in this figure how similarities between healthy controls without irritable bowel disease group together in that red shaded area, and how diversity changes or becomes less similar from one another with active or inactive irritable bowel disease. Diversity isn't only just affected by specific disease states like Parkinson's disease, but it's also influenced by other factors like diet and lifestyle too. So when it comes to our gut microbiota, we know that there are several different inputs or factors that shape our microbiome. These factors are stress, whether intentional like physical activity, or unintentional such as metabolic or psychological stressors. Pharmaceuticals such as antibiotics, we classically know will shape or even remove some of our microbes. We understand that diet is important, which we will discuss a little bit more later. And then some other additional factors to consider are geography of the host or the person, potential pollutants in the environment. I know that's talked about very frequently within the Parkinson's community. Even other lifestyle stages can contribute to individual variability. You know, from as we age over time, our microbiome may also change too. So we know that the gut microbiota can produce signaling metabolites such as short-chain fatty acids. These short-chain fatty acids can be broken down by bacteria to produce acetate, propionate, and butyrate. We'll talk a little bit more about those short-chain fatty acids later. But these bacteria can also produce inflammatory molecules such as lipopolysaccharide or endotoxin. They can even deconjugate some of those bile acids and influence cholesterol metabolism, and even produce other nitrogenous metabolites like TMAO, which you may hear a lot about within the cardiovascular disease realm. All of these signaling metabolites can communicate and influence not only the GI tract and immune system, but also the liver, the adipose tissue, the brain, of course, which we're going to highlight a lot today, cardiovascular system, lungs, and even the skeletal muscle too, we're learning more about. And there's so much interest that has developed in specifically human nutrition and the microbiome. So you can see on this graph here, I have a number of published studies that's growing exponentially each year. Documented studies have really been described as early as actually the 1400s on the benefits of bacteria in yogurt, for example, for treating diarrhea. But we had this inflection point that occurred in that early 2000s when the development of high throughput sequencing, in which we could take someone's stool sample and identify thousands of bacterial DNA present. And what was exciting for me personally is that I actually began my research career in the microbiome during this bloom of current studies that were coming out. And it's really exciting to see how far we've come in even the last decade too. And what I thought was really fascinating, I could kind of compare on this graph is when we actually look at the number of published studies on Parkinson's and the microbiome, we're truly only in its infancy of our understanding. But what have we learned, right? So how this came about was we started to notice that there was pathology occurring along the GI tract in people with Parkinson's disease. So we know people with Parkinson's have a progressive loss of dopamine neurons in that substantia nigra of the midbrain. And it's estimated between 60 to 70% of those neurons are lost when those motor symptoms appear. So that's what they refer to as that motor stage. That pathology that's found in the brain is an accumulation of Lewy bodies, so an aggregated alpha-synuclein. that are found in the neurons of people with Parkinson's. Now, there's a lot of debate when it comes to proteins in the neuroscience community like alpha-syn, but it has been postulated that this accumulation of alpha-syn may trigger loss of those dopaminergic neurons, but we can also find these proteins in other neurodegenerative diseases, so they're not very specific. And what has got us thinking more about the gut as it relates to Parkinson's is that we have actually been able to find alpha-syn pathology all along the GI tract of someone with Parkinson's. So whether that's the submandibular gland, the oropharyngeal region, the stomach even, the appendix, and of course the colon too. And when we think about those non-motor symptoms of Parkinson's disease, I know probably one of the most common ones that I'm sure plenty of my people with Parkinson's that are here on the line and those care partners too experience daily is some type of gastrointestinal dysfunction. And that's because it is fairly common in someone with Parkinson's, whether that be dysphagia. It's estimated that up to 77% of people with Parkinson's have some type of dysphagia. And we can do current treatments available, of course, would be speech therapy, surgical treatments if necessary, and then potentially even some dopaminergic medications too, if it's indicated. Gastroparesis or delayed stomach emptying is another one that's highly prevalent in about 70 to 90% of people with Parkinson's. Right now, most of the treatments that we have for gastroparesis involve dietary interventions, whether that's modifying fiber sources or fat, potentially using certain types of prokinetic agents if it's indicated, and other types of therapeutics too. And then of course, which was the focus of my research, was constipation. And that's because constipation may affect up to 70% of people with Parkinson's. And of course, we have current therapies like increasing fiber fluids, physical activity, as well as other therapeutics that are over the counter or prescribed like laxatives, stool softeners, et cetera too. But shifting back to the microbiome, so knowing that we have this gastrointestinal dysfunction that is highly common in people with Parkinson's, and knowing that alpha-syn pathology also can be found along the GI tract, a lot more researchers over the years have started actually sequencing the microbiome of different people with Parkinson's. So this image that I have here is actually a systematic review that really looked at the number of studies and trying to find any trends of specific bacteria that were altered in people with Parkinson's. So what I will highlight is that beneficial or commensal bacteria such as Prevotella, Faecalibacterium, and even Roseburia too are found to be reduced in people with Parkinson's when compared to someone without Parkinson's. These bacteria are important because they produce beneficial signaling metabolites like those short chain fatty acids I mentioned earlier. And we'll talk a little bit more about that later too. And then they've also found Parkinson's is associated with increases in other specific bacteria like Bifidobacterium and even Lactobacillus too. And these are actually known commensal microbes. And it's suspected that these might be increased due to the high prevalence of constipation. But we also have increases in pathobiomes like Bilophila and even Akkermansia too, which is which is an interesting one that I won't touch on too much right now. That might be beneficial or not, depending on how abundant it is in the GI tract. We've also had some studies published over the years looking at different markers for intestinal permeability and inflammation in someone with Parkinson's. So on the left here, I have an image here showing you measures of fecal zonulin. So what zonulin is essentially a protein or a marker of intestinal permeability. And we know that this marker is increased in GI inflammatory conditions like celiac disease, IBD, type 1, and even other autoimmune diseases. And you can see when we compare fecal zonulin in people with Parkinson's to controls, that is significantly elevated, indicating increased intestinal permeability. On the right here, I have calprotectin. So this is another marker of intestinal inflammation. It's actually very highly sensitive, but low specificity as a fecal marker for other irritable bowel diseases. But again, you can see in people with Parkinson's, calprotectin, and this is just one study, but there have been a couple others that have been published too, is significantly increased compared to those without Parkinson's, signaling that some type of intestinal inflammation going on. And so with our knowledge of these changes in the GI tract occurring in someone with Parkinson's, it's led to this earlier hypothesis about 20 years ago by Brach et al. That we call the Brach hypothesis. And so what Brach and his colleagues postulated was that alpha-synuclein pathology may actually spread from the GI tract via the vagus nerve to the brain. And this is just an image here showing that hypothesis too. There's still a number of researchers still trying to investigate this hypothesis, but this is the origination of this gut to brain spread in someone with Parkinson's disease. And we do have some recent preclinical models that do suggest this type of gut to brain spread via the vagus nerve. So this was a study that was published in Neuron. It was a fabulous study. It's a very technical paper, so I'll do my best to simplify it down for you all. But what they did was they injected mice with this exogenous preformed fibrils, or PFF is how I'll refer to it, and alpha-synuclein within the GI tract of mice. And what they aim to do is measure the amount of that pathologic or aggregated alpha-synuclein accumulation in the brain. They saw that mice with intact vagus nerves accumulated that aggregated protein, that alpha-syn, in the brain, specifically in that substantia nigra region, and they did develop PD-like symptoms. However, when these mice, when they actually performed a vagotomy to cut the actual vagal nerve, and they injected with that preformed fibrils, the mice did actually not develop that pathologic alpha-syn or PD-like symptoms. To prove that you actually need to have this endogenous alpha-syn to develop these PD-like symptoms, they actually did perform the same experiment in mice that didn't have a gene, which is the SNCA gene, to code for that endogenous alpha-syn. So ultimately, this study demonstrated that you do need aggregated Lewy bodies or that alpha-syn to develop PD-like symptoms. But it also established, again, that connection between that gut and brain axis in its development of Parkinson's disease. And this is just kind of a summary slide to kind of put together this Brach's hypothesis and the gut-brain axis. So you can see here, it's postulated that we have maybe some environmental, genetic inputs that causes maybe some immune dysregulation and inflammation, leading to a reduction in beneficial microbes, those beneficial short-chain fatty acids, and an increase in those pro-inflammatory cytokines or signaling molecules, an increase in that LPS, zonulin, and calprotectin, as well as that increase in intestinal permeability. This leads to potentially what we refer to as leaky gut, right, that you see here down at the bottom. And that communication may spread from that enteric glia within the GI tract through the vagus nerve to that central nervous system in the brain for PD-like symptoms. So this is just a theory, but this is to kind of summarize this gut to brain axis spread in someone with Parkinson's. But particularly for me, as a dietitian by training, I've always been interested in how diet may modulate this mechanism. Can we utilize dietary interventions to increase those beneficial microbes, short-chain fatty acids, and decrease that inflammatory response? And we know that the influence of diet on low-grade inflammation and intestinal barrier function is becoming more relevant to our understanding of metabolic diseases. The gut barrier consists of beneficial microbes, the mucus layer, the intestinal epithelium, immune cells. Dendritic cells, which are those purple cells that you see here on the picture, are continually sampling the lumen of the GI tract to promote immune tolerance through proliferation of regulatory T cells and mediate pathogen evasion via secretory IgA. We also have Paneth cells, which are that red cell that you see there, that can actually secrete antimicrobial peptides, or AMPs, in response to immune signaling induced by the gut microbiota. When we consume fibers and prebiotics that are added to the diet, we actually have conversion of those fibers to those short-chain fatty acids by the microbiota, which supports mucus production, by the goblet cells, expression of tight junctions, proteins to hold those epithelial cells tightly together and reduce inflammation. We know that low fiber diets are associated with a decrease in the mucus layer and reduced expression of tight junction proteins that promote intestinal permeability, leading to inflammation and further contributing to the development of metabolic diseases and potentially Parkinson's disease too. So just to give you a little bit more information on short chain fatty acids. So like I mentioned, short chain fatty acids are produced by the microbiota through fermentation of fibers or non-digestible carbohydrates. One of the short chain fatty acids that I love to talk about and a lot of microbiome researchers are very interested in is butyrate. And that's because butyrate is a major energy source for colonocytes. But despite this, it's actually the least abundant short chain fatty acid produced by those intestinal microbes through fiber fermentation. We know that butyrate producing microbes frequently rely on the cross-feeding of others, which I'll show a slide on that a little bit later. And butyrate has the ability to influence immune function and even gene transcription too. So primarily this anti-inflammatory effect. And recently, just last year, actually, researchers have looked at the relationship of fecal butyrate and how it correlates in someone with Parkinson's. So this study was a case control study where they compared someone with Parkinson's and those without. And they found that people with Parkinson's had significantly less fecal butyrate in their stool samples that you can see on the figure on the left. And when they compared that butyrate to those MDS UPDRS Part 3 scores. So for those that aren't a movement disorder neurologist, these are going to be that motor symptom severity scores that our movement disorder neurologists are testing on visits. So you can see this inverse relationship. So as butyrate decreases, motor symptom severity potentially increases too. But again, this is a correlation study. So you want to keep our interpretation of this evidence is pretty limited and preliminary at this point right now. So now we're going to kind of shift gears of the presentation and really dive a little bit deeper on dietary interventions that can modulate the gut microbiome. So bringing it back to this figure that I showed you earlier that describes those different inputs. As a dietitian, you can probably expect I've always been fascinated by how dietary interventions can modulate the microbiome to improve our overall health and quality of life. So initially, this interest definitely began with fiber, prebiotics, and probiotics, but it's now even expanding into others like symbiotics and postbiotics. And we'll discuss a little bit more on that later. But again, with these inputs of diet, like fiber, for example, that microbiota can ferment to produce those short chain fatty acids like acetate, propionate, and butyrate. And when we look at how specific diet factors influence diversity, which I mentioned earlier is important for the microbiome, this was a recent study that actually further attempted to explain these factors that influence diversity using both stool and blood samples across thousands of people. So this was over 3,400 people in this study. But what I want to do, because I know this is a very nuanced and small slide, but when we blow this image up to show some of those beneficial factors on diversity of the microbiome, you can see in these green boxes here that higher consumption of plant foods such as fruits and vegetables are associated with higher microbial diversity. And this is likely due to the high content of dietary fibers found within these foods. And when we look at our intake of fiber just as a population here in the US, you can see dietary fiber is actually frequently underconsumed here in the US. We compare intake of dietary fiber using this adequate intake that comes from the DRIs. And it's recommended that we should be consuming 14 grams of fiber per 1,000 calories. So for example, if you are someone that consumes a 2,000 calorie diet, it would be recommended to get in about 28 grams of fiber in your diet. Where this information comes from is actually its benefits on cardiovascular health and outcomes too. And when we actually look at how fiber intake changes over different age groups, you can see overall, most people are only consuming probably about 14 to 18 grams of fiber per day, or about 8 to 9 grams of fiber per 1,000 calories. And when we look at older adults, especially because a lot of our people with Parkinson's are over the ages of 60 and above, but even my young onset Parkinson's too, you guys can see both of these intakes still not meeting those fiber recommendations. And again, we know that dietary fiber can aid in both GI health, but also providing that energy source for beneficial bacteria in the microbiome. So dietary fibers, just to kind of give a brief review of that, we know are very known for their ability to promote laxation, right? We hear about it a lot when we think of constipation management. And they can even decrease the rate of absorption of cholesterol and glucose from the diet too. Dietary fibers promote microbial diversity through fermentation. Prebiotics such as fructooligosaccharides, galactooligosaccharides, and inulin are actually another class of fibers that are selectively utilized by host microorganisms to confer a health benefit. So this benefit is usually due to increased abundance of beneficial bacteria, like bifidobacterium, for example, that can lead to increases in those short chain fatty acids like acetate, propionate, and butyrate. These short chain fatty acids can actually be used as an energy source for other microbes and colonocytes, and even modulate that immune function and colonic pH too. Short chain fatty acids can increase secretion of actually gut hormones like GLP-1 and PYY. These are peptides that are related to gastric emptying, appetite and satiety, and even glycemic control. So increased secretion of these peptides via short chain fatty acids are also implicated as targets for treating other metabolic disorders too, like type 2 diabetes. And I know there's a lot of work in this area as it relates to the gut-brain axis on how increases in these peptides may also impact appetite and satiety too. And just to kind of give you a brief, quick summary of how that fermentation occurs by the bacteria. So we have this dietary fiber, which is that non-digestible carbohydrate that's seen in that green chain there. We have, and I have an example of different types of fibers that may be used. So when that fermentation occurs, we have normally a primary fiber degrader. Maybe it's Ruminococcus bromii that can break down that fiber into smaller fiber units, and even those short chain fatty acids like acetate, propionate, and butyrate. And then we have also, so cross-feeding by secondary fiber degraders. So that could be, for example, Eubacterium hallei, that can further break down those smaller fiber fragments into those short chain fatty acids like butyrate that can work as a signaling molecule on the GI tract, the immune system, and potentially that gut-brain axis too. And moving on to just talking a little bit more about, because I talked a little bit about dietary fiber, but really kind of diving into what prebiotic fibers are. So these are substrates, again, that are selectively utilized by host microorganisms to confer a health benefit. And what I really want to point out is that not all fibers actually meet this definition. We have some foods that naturally contain prebiotics like bananas, onions, garlic, chicory root, which is the source of inulin, artichokes, and even beans too. We know that prebiotics can also be added to food products, like in the form of GOS, FOS, oligofructose, chicory root, or inulin. So you might see this, for example, in different food products like fiber bars, or even lately I've been seeing this a little bit more with protein shakes and other types of drinks too. And when we look at what the recommendation is of how much prebiotics that we should be getting in our diet, we actually can look to the International Scientific Association for Probiotics and Prebiotics. So this is an expert consensus working group of researchers in this area that really came together to establish these definitions of what is considered a prebiotic, a probiotic, a symbiotic, et cetera, right? So I have this figure here just to use as a reference here, just to show you how prebiotics are different than dietary fibers and other fibers that aren't prebiotics too. But shifting gears, I've spent some time discussing that prebiotic effect of fermentable fibers. However, we know that other compounds like polyphenolic compounds, and for example, proanthocyanidins found in grapes or cranberries, can also be considered a prebiotic, and they're important for that intestinal barrier function of the GI tract. So we have bacteria like Akkermansia mucinifila that resides in the mucus layer of the GI tract and is a potential biomarker for barrier integrity. Now, I will say that, especially within the neurological and the Parkinson's disease world, Akkermansia can be a little bit more nuanced and complicated. We see in people, for example, with obesity and diabetes, for example, actually have low counts of this microorganism and associated with intestinal permeability. But it's interesting, we actually see the opposite in someone with Parkinson's, where they have this overabundance of Akkermansia in the GI tract. So there's really more work to be done in this area, and maybe it's just a signal that there's this balance occurring. But we know that Akkermansia can metabolize those polyphenolic compounds down to other phenolic metabolites as well, and that can be reabsorbed back into the liver and enter systemic circulation too. In the GI tract, they can also signal production of mucin by the goblet cells, which is used as that energy source for Akkermansia to bloom. An abundance of Akkermansia can have this bidirectional effect on stimulation of mucin secretion. And to show what it looks like of Akkermansia's work in the colon, this is just a cross-sectional image here of a mouse model, of an obesogenic mouse model that was fed a standard chow diet, so a high-fat, high-sugar diet, or a high-fat, high-sugar diet with polyphenols. So you can see at the same magnification, the mucosal thickness was drastically reduced with the high fructose, high sugar diet compared to controls. So this thickness was normalized when supplemented with polyphenols, and it was associated with a bloom of Akkermansia. So again, these investigations really need to be further confirmed in humans, right? And their relevance to improving clinical outcomes, especially as it relates to someone with Parkinson's who actually might have some overabundance of Akkermansia. But that can also have some variability there too. But I definitely predict we're going to see a little bit more of Akkermansia and polyphenols even as therapeutic targets for different disease states like people with Parkinson's. Moving on to probiotics. So probiotics you probably hear very frequently about. So these are going to be those live microorganisms that, when administered in adequate amounts, can confer a health benefit to the host. But one of the things I always want to keep in mind, especially within the Parkinson's community, is that benefits of probiotics are going to be species and even strain specific. So not all probiotics are going to function the same way. They're not going to function the same way in everyone with Parkinson's, and they're definitely not going to function the same way between different individuals too. When we think about the differences between probiotics and fermented foods, there's evidence for a health benefit, but that's not required for fermented foods. So examples of that would be yogurt, kefir, kimchi, even cheese and some breads too could be a fermented food, but not a probiotic. You might have also heard the term symbiotic, which is a combination of a probiotic and a prebiotic. But one of the things to keep in mind is that there really isn't a general recommendation that exists for consumption of probiotics. I do have this clinical guide that I'll reference here for you all. It's a website for probiotics in the US. I find it really helpful. I have the link here at the bottom where you can actually search the different types of probiotics that are available on the market in the US and actually take a look at the strain and its potential application that's been found in research too. And when we think about diet for someone with Parkinson's, what I always am really communicating right now is that we're not there yet as it relates to what's the best diet for someone with Parkinson's, but we are getting closer. So majority of research has focused on identifying risk factors for Parkinson's, and trials have shown strong results in humans unless you have a clinical, haven't actually shown strong results in humans unless you have a clinical deficiency. And so a lot of the research, especially in Parkinson's, and for my scientists on the line, I'm going to challenge you all to think about too, is we've done a lot of research on looking at single food components, but we know that we don't just individually eat one specific vitamin or protein, et cetera, each day, right? We eat a whole pattern of foods. So you really want to consider this as we move the research forward too. And just to kind of briefly share with you some of the evidence that we do have right now in the literature as it relates to probiotics in someone with Parkinson's. This was a symbiotic drink that a research group out back in Italy did in 2016, showing that a symbiotic drink increased the number of complete bowel movements in someone with Parkinson's after 6 weeks. These results were also similar in a multi-strain probiotic that increased spontaneous bowel movements after 4 weeks in people with Parkinson's. This was I believe it was an Asian, maybe Japanese group that was published back in 2021. But one of the things about these studies that you really want to keep in mind is, what does the diet look like, right, when you're adding this supplement? And when we actually take a further look at this study, we find that average fiber intake of this study at baseline, so before intervening, was about 30 grams per day, okay? So when you think about someone in the US, right, we're consuming half of that too. So it's interesting to think how these results might actually differ here in the US. We also have a new era of dietary interventions that can modulate our gut microbiota, which are called postbiotics. So this is a new definition by ISAP, that it's basically preparation of inanimate microorganisms that can confer a health benefit to the host. And I know I'm kind of getting short on time, so I'll just kind of briefly review this. But if you look at the figure here, so at the top, it's a live and active form of a common probiotic, Lactobacillus rhamnosus GG. And at the bottom, you can see it's an inactivated form. So there's been some evidence for efficacy of even postbiotics in treating conditions like infections and other disease states like IBS and even COPD too. However, we do have some limited safety studies published in this area and really no regulatory guidance from the FDA in this category. But I anticipate you may see some new research, down the line as it relates to postbiotics in the gut-brain axis even too. And then finally, we're going to end about, just talking about what about the Mediterranean diet, right? So we know that the Mediterranean diet, those health benefits, it provides antioxidants from plant foods. It acts as scavengers to help prevent or slow damage to our body cells. It also provides a high level of omega-3s from the fish and seafood from the diet that can influence brain health and function, and even dietary fiber from plant foods too. There have been a number of different studies that have actually looked at the Mediterranean diet and how that changes that microbial composition in healthy populations and some other disease states too. And it's also been associated with higher levels of those beneficial short-chain fatty acids like butyrate I mentioned earlier. And then when we think about how the Mediterranean diet might actually influence brain health, the PREDIMED trial is really that classic trial that demonstrates this. So this was done back in Spain, published in 2015. And they took older adults, so over 65 years, that were at high risk of heart disease, and assigned them to either a Mediterranean diet with extra virgin olive oil, a med diet with nuts, or a healthy control diet or low-fat diet. And they actually intervened for 4 years, which is a very difficult thing to do in research. But what they found was that a Mediterranean diet actually may slow age-related cognitive decline. And this didn't matter whether you were supplementing with olive oil or nuts, but these tended to do better than that healthy low-fat diet group. When we look at Mediterranean diet's association with Parkinson's disease, we have a couple studies showing that it's associated with lower risk of developing Parkinson's. So maybe for those in your family, this might be something to think about in terms of diet quality and risk of developing Parkinson's. And even prodromal symptoms too. So that prodromal stage is going to be before that motor stage, where a Mediterranean diet is associated with less non-motor symptoms in that early phase. Those flavonoids and flavonoid-rich foods that are found within the Mediterranean diet have also been associated with decreased mortality or risk of death in someone with Parkinson's as well. And even a small study that we did here at the University of Florida found that a 5-week Mediterranean diet altered some of those bacteria that we find that are altered in someone with Parkinson's. So for example, Roseburia and Bilophila here too. So when we put this all together, just that brief take-home point, how this works is that we really want to consume a diet rich in those plant foods like a Mediterranean diet or other prebiotics, fibers, etc. And really ensuring that dietary fiber intake is between that 25 to 30 grams per day range to increase that production of beneficial microbes and their byproducts, which is those short-chain fatty acids that can potentially modulate that gut-brain axis and brain health too. Now, in the future, we're going to definitely see this leverage of machine learning and AI and precision nutrition for treatment of Parkinson's disease. So this old framework, we took a target population, we gave them a dietary intervention, sequenced their stool samples, and we had lots of different heterogeneous outcomes. Where we're actually moving in the literature now to a new framework of inputting the microbiome, metagenomics, diet, genetics, and other clinical parameters into these machine learning models, in which we can actually use AI to predict and create precision nutrition strategies that will modify an individual's microbiome and further improve the response to a specific diet. Now, this is definitely still in its infancy, but a decade ago, this model definitely seemed like a dream, and now it's actually becoming more of our reality, and I'm really excited to see kind of where this next decade of research in the gut and brain axis takes us. So in summary, we talked about how the gut microbiome and gut-brain axis are implicated in people with Parkinson's. Diet like fiber, prebiotics, probiotics, and even the Mediterranean diet can shape that microbial diversity and metabolic capabilities of the microbiome. And that response to the gut-brain axis is going to be host specific. And so we really need, and this is a call out to all the researchers that are listening in, those precision nutrition strategies to potential differentiate some responders to these dietary interventions as well. And with that, I am happy to take any questions that you all may have. Thank you again for your time.
Thanks, Dr. Rush. I really appreciate that. That was an incredibly detailed presentation, and clearly the research in this area is evolving tremendously. So thanks for that tremendous overview of that. It seems like there's, given the technical nature of where we stand, how do we turn that into something that becomes really pragmatic for our listeners? And I think that one of the slides you showed began to get there. So yep. Part of it, and I think if I can summarize, it seems like a bottom line here is fiber. It's really something that people should be focusing on. We don't take enough of it. It greatly impacts our microbiome, and our microbiome, by giving off various chemicals, some good, some bad, certainly play a role in gut health and then impact brain health as a result of that. I mean, is that a fair assessment?
Yes, it's definitely a fair assessment, Dr. Beck. So, when I was practicing as a dietitian in the clinic, I was always educating to people that just to start with the basics, right? We're seeing the forest within the trees, that analogy. I might have butchered that a little bit, right? So we really just want to go back to the basics of diet quality, which includes increasing those fruits and vegetables, getting more of that dietary fiber in the diet, right? And making some modifications from there, right? And so that's how we can really begin to customize and provide that individual nutrition strategy, because if we don't have the basics and that foundation set up, then it's really hard to supplement or determine what's going to be the right fit for someone with Parkinson's as it relates to the microbiome and the gut-brain axis.
Fantastic. And let me just pause real quick and just let our audience know that continue to submit questions using the Q&A icon below. And if you're on Facebook, share your questions in the comments. My colleagues are organizing the questions as they come in, but a lot of them are coming in, and so we're not able to respond to all of them as much as we would. So our helpline is also a resource to answer questions that may not be answered that are still persistent as part of that. So let me just kick off with some additional questions. So I think we've established this brain-gut connection. It's both the physical one through the vagus nerve that may contribute to the development of Parkinson's disease, but it's also this less clear one, let's say through the circulatory system, where the impact of gut health can have, whether it be constipation or the microbiome itself can have for that. So a lot of people with Parkinson's are here on the line, and they're asking some questions. Like, let's just tackle a practical one as we think about gut health. People with Cinemet a lot of times experience nausea as a side effect of it. But is it all because of nausea? Can constipation lead to some of these issues? And this gets to the idea again of we talk about gut health microbiome. What can we do as part of that?
That's a wonderful question. Definitely probably the number one question I always got when I was in clinic too, as it relates to taking that Cinemet or that levodopa medication and how the influences of GI dysfunction impact it. So I actually published, I was first author on a review paper that was published earlier this year in MPJ Parkinson's Disease, which is a partner journal with the Parkinson's Foundation, that actually describes this interaction between GI dysfunction and levodopa. So there are actually a few studies that have looked at the pharmacokinetics of when we take oral levodopa, how GI dysfunction impacts those pharmacokinetics. So constipation certainly may impact the pharmacokinetics, so reducing the efficacy of levodopa. Gastroparesis, or that slow gastric emptying as well too. So for my providers that are listening in, and even my people with Parkinson's, right? When we think about when our levodopa isn't correctly working the way it should be, right? And we're not getting that good on and off time. Oftentimes when we think about diet, we always think about the protein aspect because there are potential protein interactions with levodopa. But one of the things I might want to challenge my healthcare providers and even people with Parkinson's and their caregivers is to think about the GI dysfunction and how that might also impact the pharmacokinetics of the medication too.
Fantastic. And let me just, I'm going to post a link to your paper to my colleagues and they can push it out. The great thing about NPJ Parkinson's is it's an open access journal, so anyone listening can download it without a paywall as part of that process. So great, thank you for that answer. And then, so taking to account to some of the things you were talking about today, butyrates, some of these small chain fatty acids that are playing an important role. Is the research suggesting that you take it exogenously, or is it better to spur one's microbiome to make more of it directly? Where are we headed with that?
That's a really great question. So we need more research on butyrate as it relates to how it can modulate, especially the gut-brain axis. There are actual oral exogenous forms of butyrate on the market. I know, for example, I think it's tributin is one. The challenge with taking exogenous butyrate is that the taste is very terrible because it is a short chain fatty acid. If anyone's tried exogenous ketones, for example, you might experience a similar flavor profile as well. But
it gets terrible.
Yeah. So that's been one challenge with oral butyrate, and the other challenge is how to study it too. Because when we take exogenous butyrate, I mentioned that colonocytes are epithelial cells. They're constantly rapidly turning over and producing new ones, right? When we take something exogenously, whether that's by the mouth or actually there's butyrate enemas that some studies have looked at, particularly in like a critical care population, those enterocytes in the GI tract metabolize, I think the number is around 70% of that butyrate just for those enterocytes, and the remaining amount might get back into circulation. So it's really difficult to study how much that I gave you of butyrate actually does end up in circulation. Usually you need a more sophisticated method like radioactive labeling to really investigate that type of mechanism too. So that's why you've seen the literature really focus on these other interventions to increase butyrate production. So this indirect effect on the gut-brain axis, like fiber, prebiotics, and probiotics too.
Yeah, because it seems like you mentioned it as a food source for other bacteria in your gut, and they rely on it. So it seems like it could be beneficial for that to help restore a microbiome. And it leads to my next question. We talk about the microbiome. How stable is it? I mean, so using dietary fiber as an example, we're clearly not taking enough of it. And so if you take more of it, I mean, how long would it take to see a change? And then I guess the other point you bring home in your talk is not all dietary fiber is the same. So yeah. How do you do that? Because like one of your things you mentioned are bananas, which for a person with Parkinson's is not exactly the fruit they reach for first because of the constipation factor.
Yeah. So, and it's a really great question too. So, how to kind of how to manage this. Actually, Dr. Beck, can you repeat the first part of the question? Because I think I'm
Yeah,
blanking on it a
yeah,
little bit. Yeah,
I threw a lot in there, so no problem at all.
that's
So
okay.
how stable is the
Yes,
microbiome at all?
stability.
So yeah, because it seems like, especially when we take probiotics, you're hoping to change, but I think, and this is my terrible analogy, but I think about, if you have an unhealthy microbiome, it's like your yard covered with weeds. And if you're taking probiotics, it's like throwing grass seed down. I still have weeds. And what do I need to do is, clear house. And do you have to do
Yeah.
that in order to really get probiotics to work? Do you need to take antibiotics in order to like purge your gut and then start fresh? Or what are people thinking these days?
Yeah, that's I really like that question. So in terms of stability of the microbiome, yeah, our microbiome is very stable when we look at it over time, right? It can change very in the short-term period. So for example, if we give someone a short-term antibiotic, or if we give someone a probiotic, right? We can see some short-term changes in that resident microbiota. But over time, especially, as we come off the medication and there's some type of a washout period, now you're back in your normal environment, you're eating the similar foods again, right? You tend to see that people tend to move back to their resident microbes, right? And so when it comes to changing the microbiome, that's been one of the most difficult challenges that researchers have had. That's why particularly fiber can be a great strategy, because you're getting that broad spectrum changes, because those fibers aren't specifically targeting one bacteria, right? They can be broken down by multiple types of bacteria that other bacteria that are resident to your microbiome can use for cross-feeding, et cetera, right? When we give a probiotic, we're only increasing abundance of one bacteria in the community in the hopes that it creates a new niche, and it takes resident in that microbiome. But often what we see is that when you take a probiotic, if you miss a couple of days, your microbiome kind of goes back to normal too. And so there have been some researchers that have been looking at, do we need to do maybe some like short-term antibiotic treatment and then do a dietary intervention as well, and whether that's a probiotic or another type of therapeutic too, and see how that changes the microbiome and can that create stability. We're definitely in its infancy there. Even fecal transplants too. So I know a lot of people with Parkinson's, there's been more interest in the community there, right? Because again, you're changing someone's microbiome, right? But even with fecal transplants, you still may get some changes back to your original residential microbiota over time. So the question becomes, do you need to do multiple fecal transplants? So again, definitely in its infancy. But I love the analogy that you made as it relates to, the seed starter over the weeds, right? Because you're right. That's a really good analogy for it too.
Yeah, thanks. And I think also about I could understand why it's hard to establish the change, because you really need to change the conditions for the bacteria to grow. So you can take the probiotics, that's great. And unless something fundamentally else changes, then it may not help. And perhaps that could be fiber, right? Because that is really seems like a good food source for a lot of the stuff. So when we talk about fiber then, and I know we've just have a few minutes left, how do you measure that like on a practical level? I mean, you go out to eat, hopefully, and you're trying to have this, not everything has a labeling on it. And so how do you make these choices to do that? Are there resources available perhaps to you could recommend as part of the process?
Yes. So in general, your high fiber foods are going to be plant foods. Okay. So your fruits, your vegetables, beans, whole grains, even too, right? And sometimes other types, and then you could also have additives as well. So if you don't have a label, those are going to be the things that I'm looking for to get more fiber in my diet, right? I'm going to be looking for plant foods, and it's recommended by the USDA. So the MyPlate, you can actually go on to choosemyplate.org, where you can actually see more resource, like very simple nutrition education resources. And what they recommend is actually using the plate method. So filling half your plate with fruits and vegetables, okay? And that's one way as a visual strategy to get more fiber in the diet. But of course, if you do have labels, available to you. You can take a look for labels as well. It will actually have a daily value percentage next to that fiber. So one of the questions is, well, how do I know if this is a high fiber food? You can look to that daily value, and if it's over, I believe it's 10, it's considered a good source of fiber in the diet.
Okay, awesome. Well, I think we've reached the end of our time. I appreciate all the questions that have come in as part of that. So, again, thank you, Dr. Rush, for offering your knowledge and time today. And talking about gut-brain connection, and big thanks to everyone who's joined us today. So we, again, had a lot of questions, not able to answer them all. So if a question was not answered, please call our helpline, 1-800-4PD-INFO. And I also want to just highlight that we've completed a good chunk of our expert briefings today. So we've got one left, and it's going to be one that's not to be missed on Wednesday, November 8th. And so it's about hallucinations. This is something that people are concerned about, and some people have at various levels. So I think it'll be a great one to register for and to learn about as part of that. So we are here for you, as always. Our website, our helpline, via telephone or email. Those are good ways to get in contact with us beyond questions regarding this gut-brain axis, other issues that you may be facing as part of that. But before you go, I just want to, we're in the Zoom world, and it's just going to go to black when we end this webinar, but a survey will appear afterwards, and we really want your input on this. We use that survey to provide feedback to our speakers. We use that survey results to provide feedback on our program as a whole. So please just take a few minutes and complete that survey when it appears. Otherwise, we look forward to speaking with you again come November, and we'll take care.